Now that we've covered 3.3, the cell membrane, we can now talk about some movement of particles. These movements can be called diffusion and osmosis. Osmosis is going to be something that we learned about with that naked egg lab that we did. Materials are going to move across membranes because of concentration differences. So all that's really saying in this particular picture here is that outside the cell, there's a larger concentration. There's more of the blue circles, which I think we're trying to, I guess we can assume is water. There's more water outside the cell than there is inside the cell. So passive transport does not require energy input from a cell. So anytime anything's passive, it's happening without a whole lot of effort. So passive transport does not require energy input from the cell. Molecules can move across the cell membrane through passive transport. So when this happens, it's just kind of easy peasy. It just moves very easily across. There can be two types of passive transport. There's diffusion and there's osmosis. So imagine with diffusion, if you had a large vase full of water and it's just clear, plain old water, and you drop a few drops of food coloring into that water, what's going to happen? Well, the food coloring is not going to stay as several large blobs of red food coloring. It's going to eventually diffuse throughout the water and make that entire vase full of water, a light red color. So that's one type of passive transport. Osmosis is a type of passive transport that we got to witness when we did the naked egg experiment. Oh, I didn't even know this is the picture they used. Molecules diffuse down a concentration gradient, and this is somebody dropping a few drops of food coloring into a beaker full of water, much like what I was just talking about. Osmosis is the diffusion of water molecules across a semi-permeable membrane. So again, osmosis is going to be the diffusion of water molecules. So anytime you're talking about osmosis, it's water molecules, and it's going to be moving across a semi-permeable, so that means some things get let through and some don't, semi-permeable membrane. So diffusion and osmosis, again, are a passive transport. There are three types of solution. There is isotonic. And isotonic is going to mean that it's a solution is isotonic to a cell if it has the same concentration of solutes as the cell. So it's the same on the outside as it is on the inside, basically. I am just trying to move this for you so you can see this a little bit better. There we go. So if a solution is isotonic to a cell, it has the same concentration of solutes, the dissolved materials, as the cell. And equal amounts of water are going to enter and exit the cell, so it stays the same. So this is a picture of a red blood cell here with the number 1, and this is in an isotonic solution. So this red blood cell is happy. He gets to stay shaped like a donut, gets to do his red blood cell thing and transport nutrients and waste materials throughout your body. The second picture, labeled number 2, is a hypertonic cell. It's hyper, it's over, right? Just like if you were hyperactive, you're like overly active. And a hypertonic solution has more solutes than a cell. So again, that hyper, it's over. So a hypertonic solution has more solutes, more dissolved materials than the cell itself. Overall, more water is going to exit the cell in a hypertonic solution, causing the cell to shrivel up and it could even die. So again, hypertonic is going to have more solute in the solution than inside the cell. So water is going to leave the cell and go out into the solution, and that's going to cause it to shrivel up and potentially die. So in the case of our naked egg experiment, this would be the eggs that were inside of the Karo syrup. And then a hypotonic solution, which is labeled number 3 here, is a solution that has fewer solutes than the cell. So hypo is lower. So a hypotonic solution has fewer solutes than the cell. And in this case, more water is going to enter the cell than in the hypotonic solution. And this is going to the cell then is going to expand and it could even burst. So these are all pictures of red blood cells, just in different situations. Number one is isotonic, so it's happy. It's the same on the outside as it is on the inside, and water is just flowing back and forth across. Nothing's really changing. It's wonderful. So it's a nice, happy red blood cell. And number two, it's hypertonic solution. So the solution itself is hypertonic, so it has more solute, which means in this case, it could cause the cell to shrivel up and even die. So again, that's like the Karo syrup when we did our egg experiment. The hypotonic solution, the solution itself has fewer solutes than the cell, and so water is going to pour into the cell, causing it to swell up and possibly even burst. This example would be our eggs that we left in the distilled water. Some molecules can only diffuse through transport proteins, so they can't just bebop their way across that phospholipid bilayer. They need transport proteins to help them across. Facilitated diffusion is diffusion through transport proteins. So in this picture here, whatever our little blue circles are, need some help to get across the phospholipid bilayer. And to get that help, they're going to use transport proteins, which do exactly what they sound like they would. They're proteins. They're these areas that are made out of protein, and they are going to allow passage of material through them. And that brings us to the end of diffusion and osmosis. I hope this, combined with your egg lab